The overall goal of this procedure is to incorporate GAG genes derived from subtype C HIV-1 infected individuals into a replication-competent HIV-1 plasmid in order to assess the effect of GAG sequence polymorphisms on HIV-1 replication capacity in vitro. This is accomplished by first amplifying the GAG gene from the patient's plasma. The second step is to clone the patient-derived GAG sequence into the replication-competent infectious molecular clone MJ4. Next, the GAG MJ4 chimera plasmid is transfected in order to generate infectious viral stocks and titered to assess infectivity on an indicator cell line. The final step is to measure in vitro replication capacity in the GXR25 T-cell line. Ultimately, a radio-labeled reverse transcriptase assay is used to quantify virion production over time in order to visualize viral replication kinetics.
The method we're going to describe today can help answer key questions in the HIV field. These include defining the determinants of HIV replicative fitness and understanding how HIV fitness affects long-term pathogenesis. Describing the methods today will be Jessica Prince and Dan Claiborne, two of my graduate students.
To begin this procedure, reverse transcribe cDNA from RNA and amplify the first-round DNA products using reverse transcriptase and a thermostable DNA polymerase in a one-step RT-PCR. Then, using 1 microliter of the first-round PCR amplification as the DNA template, perform a nested second-round PCR amplification. Next, add 3 microliters of 5x loading dye to 5 microliters of the second round reaction volume. Run it at 120 volts on a 1% agarose TAE gel containing a UV fluorescent DNA stain until the bands are resolved. Then, visualize the 1.6 kb bands on a blue light illuminator. After that, run the remaining 45 microliter reaction volume on a 1% agarose TAE gel containing the DNA stain. Excise the appropriate bands with a clean razor blade and combine 3 positive reactions per individual. Subsequently, extract DNA from the gel slice using a gel purification kit and elute it in the nuclease-free water. Freeze the product at -20 degrees Celsius for use later. In this procedure, after amplifying the long terminal repeat 5' UTR region of the MJ4 plasmid, visualize the 1.3 kb PCR products on the illuminator. Excise the positive amplicons, then purify and freeze them as previously described. Next, create the fused MJ4 LTR GAG amplicons via Splice Overlap Extension PCR. Visualize, excise, purify, and freeze the 3.2 kb long amplicon. Now, digest 1.5 micrograms of MJ4 plasmid and 1.5 micrograms of purified LTR GAG PCR product with BCL1 restriction endonuclease for 1.5 hours at 50 degrees Celsius. Subsequently, add 1 microliter of NGOM4 to the digestion reaction and incubate it at 37 degrees Celsius for 1 hour. After that, add 5x loading dye to the restriction digest reactions. Slowly electrophorese the total volume on a 1% agarose TAE gel containing a DNA stain for 1 to 2 hours at 100 volts. Visualize, excise, and purify the indicated bands for cloning as previously described. Next, prepare the ligation reactions using purified LTR GAG insert and MJ4 vector DNA at a 3:1 insert to vector ratio. Incubate the ligation reactions overnight at 4 degrees Celsius. Then, transform JM109 chemically competent bacteria with ligation products, spread on LB agar plates with 100 micrograms per milliliter ampicillin. Grow transformed bacteria at 30 degrees Celsius for at least 22 hours. To confirm cloning fidelity, cut the mini-prep DNA with NGOM4 and HPA1 restriction enzymes in a double digest at 37 degrees Celsius for 2 hours. Afterward, analyze it on a 1% agarose TAE gel. Calculate the volume of virus needed from each stock based on the IU per microliter titer from a TZM-BL cell assay in order to infect 5 × 10 to the 5th GXR25 cells at a multiplicity of infection of 0.05. Be sure to wear proper personal protective equipment for working with replication-competent HIV, which includes a full gown, face mask, and double gloves. On the day of infection, remove virus stocks from the -80 degrees Celsius freezer and thaw. Next, dilute the virus in CRPMI media to a volume of 100 microliters in order to achieve a 0.05 MOI. Add it to the V-bottom tissue culture treated 96-well plate, including both a positive and a negative control in each set of replication experiments, and set up the plate such that every other column is blank to limit cross-contamination between wells. Afterward, count the GXR25 cells using an automated cell counter and calculate the number of cells needed in total for all infections. Aliquot the required volume into a 50 milliliter conical tube and centrifuge to pellet the cells. Then, aspirate the media carefully and resuspend in CRPMI at a concentration of 5 × 10 to the 5th cells per 100 microliters. Pipette the cells into a sterile trough and mix thoroughly. Subsequently, using a multi-channel pipette, add 100 microliters of cells into each well of the 96-well plate containing the diluted virus and mix thoroughly. After that, add 2 microliters of 5 milligrams per milliliter solution of polybrine to each well and mix the cells thoroughly. Then, incubate at 37 degrees Celsius in a tissue culture incubator with 5% CO2 for 3 hours. In order to wash the infected cells, centrifuge the 96-well V-bottom plate to pellet the cells. Then, carefully remove 150 microliters of the medium and replace with a fresh 150 microliters of CRPMI without disturbing the cell pellet. Repeat the procedure 2 more times to sufficiently wash the cells. After the last centrifugation and the addition of 150 microliters of CRPMI, resuspend the cell pellet with a multi-channel pipette. Add the mixture from each well to 800 microliters of CRPMI in a well of a 24-well tissue culture plate. Then, place the plate in a 5% CO2 tissue culture incubator at 37 degrees Celsius. Every 2 days, transfer 100 microliters of supernatant from the surface of the culture well to a well of a 96-well U-bottom plate and freeze for subsequent analysis using the RT assay. Then, resuspend the cells thoroughly and remove half of the remaining volume to prevent cell overgrowth. After that, restore the original volume by adding 550 microliters of fresh CRPMI to each well. In this procedure, add 1 to 2 microliters of 10 millicuries per milliliter of DTTP and 4 microliters of 1 molar dithiothiotol to each 1 milliliter aliquot of RT master mix. Carefully mix each 1.5 milliliter microcentrifuge tube of RT master mix, DTT, and DTTP with a 1000 microliter pipette and transfer to a sterile trough. Next, dispense 25 microliters of labeled RT mix into each well of a 96-well thin-walled PCR plate. Add 5 microliters of each supernatant to the PCR plate containing the RT master mix. Then, seal the PCR plate with an adhesive foil cover and incubate for 2 hours at 37 degrees Celsius in a thermocycler. After incubation, make small holes in the foil cover using a 200 microliter multi-channel pipette. Mix the samples 5 times. Transfer 5 microliters of each sample to DE81 paper and allow all the samples to air dry for 10 minutes. Place the blots in separate washing containers. Subsequently, wash the blots 5 times with 1x SSC. Then 2 times with 90% ethanol for 5 minutes per wash, before letting them air dry. Once dry, carefully wrap the blot in Saran wrap and expose it to the phospho screen in a tightly sealed cassette overnight at room temperature. Once ready, analyze the phospho screens with a phosphorimeter and quantify the radioactive transcripts. Shown here are the representative gel images depicting the electrophoretic separation of the various PCR amplicons necessary to generate a patient-derived GAG amplicon capable of being cloned into the MJ4 plasmid backbone. This representative gel image depicts the electrophoretic separation of restriction digests for cloning patient GAG genes into MJ4. And this representative gel image depicts the electrophoretic separation of restriction digests of purified GAG MJ4 chimera plasmid DNA. This graph shows the reproducibility of the replication assay over time in the GXR25 cell line. The same GAG MJ4 chimeric viruses were used to infect GXR25 cells in 2 independent experiments performed approximately 1 year apart. Replication scores were generated by calculating the slope of log-transformed DLU values and normalizing that slope to wild-type MJ4. The 2 independent measurements are strongly correlated and highlight the reproducibility of assays performed at different times and with cells at different passages.
This technique paved the way for researchers in the field of HIV/AIDS to explore the viral determinants of pathogenesis in acute subtype C infection, the most prevalent subtype worldwide.
Working with replication-competent HIV-1 can be extremely hazardous. All steps involving live virus should be performed in a BSL-2+ facility. All surfaces should be regularly decontaminated and double gloves worn at all times.